Bismillahirrahmanirrahim. This video will cover the acute inflammation from phagocytosis till the termination of the inflammatory process. These are the references for this video. The learning objectives for this video are to describe the step of phagocytosis, that are recognition, engulfment, and intracellular killing. To explain the role of reactive oxygen species and lysosomal enzymes in microbial killing, to discuss the importance of NADPH oxidase system and the consequences of its dysfunction, for example, chronic granulomatous disease, to identify the key anti-inflammatory cytokines such as interleukin 10, transforming growth factor beta. They help to resolve the inflammation and to describe how inflammation contributes to the pathogen clearance and tissue repair. In the last video, we have seen that the inflammation is a sequential process. This acute inflammation starts, that is the initiation step, and there is activation of various soluble mediators, and there is recruitment of additional inflammatory cell to this milieu. Then the amplification of this process depends on the extent of the injury of the cell and or the tissue, and there is activation of some mediators such as kinin and complement system. Then the infective agent or the inflammatory agent or the damaging agent that is causing the damage, it is brought under the control, and there is a start of enzymic digestion, phagocytosis, reduction and elimination of the foreign material, and then the damaged tissue component are also removed and debris is cleared. Finally, there is the termination of this acute inflammatory process that allows repair and return to normal physiological function. A scar may form if the injury is severe. If this infective or inflammatory agent or destructive agent cannot be eliminated, the process takes the form of chronicity, that is termed as chronic inflammation. In the last video, we have seen that how the inflammatory process starts. There is recognition of the foreign material. The there is uh, there is some vascular event that occur. Then there are some cellular events that occur during the inflammation. The leukocyte that is traveling into the blood vessel in the lamellar flow, when the flow is turbulent, it comes toward the vascular wall, it attaches to the vascular wall and transmigrated from the vascular wall through the process that is termed as diapedesis, that is under the control of various adhesive molecule. Then they reach to the area where this uh, chemotactic stimulus was present. Here there is recognition of offending agent by toll-like receptors and other receptors, and there is activation of these leukocyte to phagocytose and destroy this offending agent. When leukocyte come into contact with any pathogen or any necrotic cell or some foreign particle, they are activated, and there is expression of toll-like receptors, such as these toll-like receptors are actually the pattern recognition receptors. Then there is expression of G protein coupled receptors. Opsonins receptors are also expressed, that are responsible for engulfment of opsonized particles. Cytokine receptors are also expressed, and these cytokine receptors respond to inflammatory mediators such as interferon and interleukin. Then the intracellular signaling pathway in leukocyte activation is also uh, in place, and there is increase in cytosolic calcium concentration. There is activation of protein kinase C that regulates cell survival and inflammatory mediated production. Then there is activation of phospholipase A2 that catalyzes the release of arachidonic acid that lead to formation of many of its metabolites that function in the inflammatory process. Then there are functional responses of activated leukocytes. These are phagocytosis actually, and there are steps of the phagocytosis. First, the microbe is recognized, it is attached, it is engulfed by this phagocyte, and then there is intracellular killing by the production of some specific substances that uh, that do their function, that do their part of the display when the endosome is connected with the lysosome and lysosomal enzymes are liberated into this area. Then there is this intracellular killing is it is completed by the production of reactive oxygen or reactive nitrogen species or release of lysosomal enzymes or proteases. Then there are other leukocyte responses that is the degranulation and release of lysosomal content, degranulation, production of various inflammatory mediator, and the finally neutrophil commits suicide by extruding all its content within the circulation. There is formation of neutrophilic extracellular traps. So all these steps are well controlled, but whenever these this inflammatory response or activation is very much increased, this will, instead of repairing the body tissue, it is going to further damage the tissue, as happens in autoimmune disease and chronic inflammatory processes. This picture is showing the activation of the leukocyte by the mechanism we just discussed. There is recognition of microbes and mediators. Here, this recognition of microbes and mediators such as peptides that are N-formyl methanol, the various chemokines and lipid mediators, they are recognized by specific receptors, and as a result the cell respond. There are changes that occur in the cytoskeleton, and also there is signal transduction that result in increase in integrin avidity, and it results finally in addition to the endothelium, while chemotaxis will result in migration into the tissue. Then the microbes are also recognized by toll-like receptors that are pattern recognition receptors, and they produce mediator from these cell membrane lipids, phospholipids, and these mediators are arachidonic acid metabolites, and various cytokines are also liberated, and this is responsible for amplification of the inflammatory reaction. Finally, there is presence of phagocytic receptor, and the microbes are recognized by these receptors they are internalized, phagocytosed, and also there is reactive oxygen species formation and lysosomal enzyme liberation that result in microbicidal activity, and finally the microbe is killed. The neutrophils and macrophages both are phagocytes. The neutrophils, they are derived from the bone marrow and they are the myeloid precursors, while the macrophages are basically monocytes that are differentiated into macrophages into the tissue. In the circulation, they are monocyte, while when they reach to the tissue, they differentiate into macrophages. In the all over the body, these macrophages in the tissue are present, and they make separate reticuloendothelial. They are part of reticuloendothelial system of the body. The neutrophils are short-lived. Within 6 to 4 hours, 24 hours, they die through the apoptosis after their action. While the macrophages are long-lived, and whenever they are differentiated into the tissue, they remain there for months or days to the months over there. The neutrophils, they circulate in the blood and they are recruited rapidly to the site of the inflammation, while these macrophages resides in the tissue as resident macrophages, or monocyte can also go to the tissue and then differentiate into macrophages. These are monocyte derived macrophages. Primary function of the neutrophil is the rapid response to acute infection or injury, phagocytosis, and microbial killing, while the macrophage is responsible for long-term response. There is phagocytosis, antigen presentation, and tissue repair that are the other primary functions of the macrophages. They destroy the offending agent. Neutrophil destroy through the formation of reactive oxygen species, lysosomal enzyme, or neutrophil extracellular traps, while the phagocytosis is a process which occur by the which macrophages follow. There is development of reactive oxygen species, nitric oxide production, and cytokine secretions also in abundance from these macrophages. Then neutrophils, they are the first line of the defense. They are a rapid response to the offending agent, but they are limited in duration, while the macrophage is a delayed response. It has the capacity to modulate with adaptation, and it leads to chronic inflammation. While the inflammatory role of the neutrophil, it promotes acute inflammation, releases inflammatory mediators such as interleukin-1 and tumor necrotic factor alpha. While the macrophage, they regulate both the pro-inflammatory and anti-inflammatory response. There are classical or they can be alternate type of the macrophages. They can be pro-inflammatory or anti-inflammatory, and also they are responsible liberation of growth factor for the tissue healing. The classic example of the neutrophil dominant inflammation are acute bacterial infection, abscess formation, pus production, pneumonia, or appendicitis. While the macrophages dominant inflammation can be exemplified by chronic infections such as tuberculosis or leprosy, and chronic inflammatory diseases such as rheumatoid arthritis, systemic lupus erythematosus, etc. This table is from Robbins and it is differentiating among the uh, neutrophils and macrophages. So as we have seen earlier, origin is bone marrow for the neutrophil, while for both of these the origin of is from the bone marrow, but some of the macrophages are resident of the tissue, and sometimes the monocyte they transform into the macrophages when they reside into the tissue. So neutrophil has got a shorter lifespans as compared to the macrophages that has got a lifespan of even weeks to months. The tissue resident macrophages can live there for years. So the differentiating feature that are the result of responses to activating stimuli. In neutrophil, the reactive oxygen species form rapidly and they are induced by assembly of phagocyte oxidase, while in macrophages this respiratory burst or this induction is less prominent. The nitric oxide is present in low levels or not in the neutrophils, while there is abundance of this nitric oxide that is introduced by inducible nitric oxide synthase enzyme in macrophages. Degranulation occurs as a major response in neutrophil, while in macrophages degranulation is not a prominent response. Cytokines have got a low level or none in the neutrophil, while there is a major play of the cytokine in the macrophage. Then net formation or neutrophilic extracellular trap formation is induced in the neutrophil only, while the secretion of lysosomal enzymes outside is prominent feature of neutrophil, while it is a less prominent feature for macrophage. The phagocytosis can be defined as ingestion by eukaryotic cell of large, usually 0.5 micron insoluble particle and microorganism. It is a sequential step, three-step process in which there is first recognition and attachment of the microbe, then there is engulfment of the microorganism, and third step is killing and degradation of the microorganism. So first they attached to the cell surface, cell membrane by specific receptors. These can be mannose receptor, scavenger receptor or opsonin receptor. In the second step, that is engulfment, the microbe is taken into the cell by zipping up of the cell phagocytic or cell membrane around it. Then a phagosome is formed with ingested microbe. This phagosome attaches with the lysosome, that is bag of many enzymes, and these phagosome and lysosome fuses and form phagolysosome. There is oxidation, and by the action of reactive oxygen species and nitrous oxide, these are degraded. So lectin, scavenger receptor, and other receptors are the receptors that are involved in the recognition. Macrophage mannose receptors are there, and they are basically they have got selective tendency, they recognize selectively the microbial cell wall. The microbial cell wall has got mannose and fucose as glycoprotein and glycolipid. So these lectin, they selectively recognize these microbial cell wall, while they do not recognize the mammalian cell wall, because mammalian cell membrane has got the sialic acid and N-acetyl galactosamine. So there is selective recognition of the cell by these macrophages, foreign invading cells by these macrophages. Then there were these scavenger receptors. These scavenger receptors were originally known as molecules that bind and mediate the endocytosis of the oxidized or acetylated LDLs that no longer interact with the conventional LDL receptors. So they take away these, but later it was recognized that macrophages integrin such as MAC1, that is also known as CD11B CD18, is also responsible for the engulfment of microbe. Then there are specific receptors for the opsonized microbe. These are immunoglobulin antibodies. The microbe is opsonized by some material. These can be IgG antibody, C3 breakdown product, or mannose binding lectin. So this table is summarizing the first step, the recognition step, in which the uh, cell is going to identify and bind the target particle. The receptors are mannose binding receptor that bind mannose and fucose on the microbial surface. There can be a scavenger receptor. They bind oxidized LDL and microbial component. Integrins, MAC1, CD11B, CD18. They bind microbe and complement coated particle. Then there are opsonin receptors, specific opsonin receptors that bind the IgG, C3b, and mannose binding lectin. So if there is deficiency of complement or IgG, there is impaired recognition of the microbe that will lead to recurrent infections in the body. So after the recognition, when the cell receptor has recognized or has identified the invading agent, then there is the process of engulfment. So this process of engulfment needs uh, specific structural changes within the cytoskeleton. There is the extension of cytoplasmic pseudopores that flow around this particle, and this area, this recognized area, it is separated from the surface and is internalized by the various structural changes within the cell cytoskeleton. So many receptor integrated or initiated signals are necessary for these membrane remodeling and cytoskeletal changes. And phagocytosis is dependent on the polymerization of the actin filament over here. And if there is any defect in these polymerization of the actin filament, it this phagocytic process can be impaired. So there is a phagosome formation. Then this phagosome is taken into the proximity of a lysosome. They merge together and form a phagolysosome. This picture is showing the process that occurs during the formation of phagosome. Here you can see first there is microbial coating by opsonin, and this microbial coated opsonized bacterium or particle is recognized by immune receptor tyrosine based activation motif. Over here this is recognized, and uh, as soon as it is recognized, there is clustering of the receptor occur, and this clustering of the receptor, this activates the intracellular signaling process, and as a result of intracellular signaling process uh, and phosphorylation by the action of tyrosine kinase, there is polymerization of the actin filament within the cell. So here these actin filaments are polymerized around this foreign agent or microbe. All these actin filaments are polymerized and they form a pit that engulf this microorganism. So the engulfment is the process by which this microorganism is taken after recognition. It is taken within the cell. There is phagocytic membrane that zips around it, and it forms after the polymerization of the actin filament within the cytoskeleton. It forms a phagosome, and this phagosome contains the ingested particle. Here this lysosome is attached with this phagosome, and phagolysosome is formed. So this engulfment is a process by which there is internalization of the bound microbes and phagosome formation. There is first the cytoplasmic pseudopods that are formed around the microbe, and they form a phagosome, and this phagosome then encloses the particle. After that, the phagosome lysosome fusion occurs, and phagolysosome is formed, where the degradation of the particle occurs due to various processes. If this lysosome failed to fuse with phagosome, it it will result in recurrent bacterial infection, and the condition is termed as Shidiak-Higashi syndrome. So this picture is describing the events that are occurring subcellularly. First, there is opsonized particle, or this C3b coated particle is recognized by cell surface C3b and Fc receptor, and when they are recognized, they recognize the microbe. This microbe is internalized, and this internalization is accompanied with the actin polymerization and pseudopod formation around the microbe, and there is complete engulfment of this foreign agent that now fuses that now forms phagosome, and this phagosome fuses with the lysosome, and lysosomal granules along with phagosome, they uh, in this structure, multiple processes occur with the help of lysosomal enzyme and oxygen radicals that are released in this area. There is killing and degradation of the invading agent. Now the intracellular killing and degradation, how this process occurs. The starting point in this step is the activation of oxidases. These oxidase enzymes are present in the cell, in the cytoplasm, and in the cell membrane. There are about 7 proteins, at least 7 proteins, they make up these enzymes, and they are present in inactive form. When they combine and assemble on the surface of the phagosome, there is oxidation of already reduced NADPH into NADP, and this instantly oxidizes, uh, this converts the oxygen into superoxide anion. This superoxide anion readily converted into hydrogen peroxide. All these reactions are happening within the phagolysosome. In the neutrophil, this after the engulfment of the particle, there is a rapid activation of NADPH oxidase. This is termed as oxidative burst in the neutrophil. So here the superoxide anion is formed that is converted into hydrogen peroxide. This hydrogen peroxide is formed within the phagolysosome. Now, myeloperoxidase enzyme from the primary granule of the neutrophil, by this enzyme action, this hydrogen peroxide is converted into hypochlorous acid in the presence of chlorine, and in the presence of iron, it is it also forms hydroxyline. Both of these substances have got strong bactericidal activities. So whenever these oxidated reactive oxygen species, they are released outside the cell, they also destroy, or they start the injury of the surrounding cell. This is the basis of the injury of the cells that accompany acute inflammation. So there are many antioxidant systems or antioxidant mechanisms are present in the cell that prevent, or they prevent this injury by these free oxygen or nitrogen radical. The protective mechanism are superoxide dismutase, and this can be found or activated in variety of the cell types. There can be presence of enzyme catalase that inactivates the hydrogen peroxide. Glutathione peroxidase is another powerful hydrogen peroxide detoxificant, and the copper containing plasma protein cerebroplasmin and the iron free fraction of the plasma transferrin are the basic protective mechanism that prevent the damage by the free radicals. Then if there is any inherited deficiency of components of the enzyme phagocyte oxidase, that is composed of 7 proteins, and these are present in various sites in the cell. So this immunodeficiency may result in a chronic disease that takes the form of granulomatous inflammation, the chronic granulomatous disease. Nitrous oxide synthase, or NOS, is an enzyme. NOS is an enzyme that forms a soluble gas, that is nitrous oxide, by the action from the arginine. There are three types of this enzyme, uh, nitrous nitrous oxide synthase. Okay, three types. So number one is inducible, second is endothelial, and neuronal. So these endothelial and neuronal are not concerned with the inflammation. The inducible type of this nitrous oxide synthase, that is present in the uh, macrophages more commonly, and to some extent more pronouncedly, and to some extent in the neutrophil they are also present. These are responsible for the formation of the soluble gas, the nitrous oxide. And this nitrous oxide is in this enzyme is induced by the action of some cytokines such as interferon, especially the gamma interferon, or some microbial products. So uh, the free radicals that form during this phagocytosis inside the superoxide anion that is formed in this phagolysosome, whenever this is formed, it also interacts with nitrous oxide and forms peroxynitrite. Peroxynitrite, and this peroxynitrite is another nitrogen dry free radical, and it similarly, as reactive oxygen species, it attack and damages the lipid, protein, and nucleic acid of the microbe. And their role in the vascular reactions of acute inflammation is not certain. So the component for the degradation and destruction of the microbes and other debris in the cell are reactive oxygen species, reactive nitrogen species, and lysosomal enzymes. During inflammation, body generates reactive oxygen species, and these are meant to kill pathogen. However, when these reactive oxygen species, when they are produced in excess, they will damage the host tissue. To counteract this action, the destructive action of these reactive oxygen species, there is presence of antioxidant mechanism, and these antioxidant mechanisms are present in the serum, tissue fluid, and host cell, and they are there to neutralize the reactive oxygen species, and hence they prevent the oxidative damage. One of these is superoxide dismutase that converts superoxide anion into hydrogen peroxide rapidly, and it is found in many of the cells. Then catalase speedily breaks down hydrogen peroxide into water and oxygen, and it is present in peroxisomes. Glutathione peroxidase, it detoxifies hydrogen peroxide and lipid peroxides using glutathione, and it is found in cytoplasm and mitochondria. Ceruloplasmin is another antioxidant mechanism that is a copper containing enzyme, and it neutralizes the reactive oxygen species and assists in iron metabolism. It is present in serum. Transferrin binds free iron, and hence it prevents the Fenton reaction that generates hydroxyl ion, and it is also present in serum. If the reactive oxygen species exceeds the antioxidant capacity, there is oxidative stress on the cell, And this oxidative stress will contribute to chronic inflammation and other diseases like atherosclerosis, neurodegenerative disorders, and cancers. So, the nutritional supports with the help of antioxidants such as vitamin C, vitamin E, and selenium can be given to enhance or support the body's defense against these oxidative damage. Major phagocytes of the body are neutrophils and macrophages. Neutrophils contain three types of the granules. One, the largest one, are termed as primary granules or azurophilic granules. These azurophilic granules contain many enzymes. One of such enzyme is myeloperoxidase that generates HOCl or hypochlorous acid for the killing of the bacteria. Acid hydrolysis and neutral serine proteases are present in abundance. For example, elastases and cathepsins, and they are responsible for digestion of macromolecule. Lysozymes and phospholipase A2, they break down bacterial cell wall and biological membranes. So these azurophilic granules enzymes, they direct microbial killing. They are important in tissue debridement and activation of inflammatory response. One of the important clinical entity is deficiency of myeloperoxidase enzyme that results in impaired killing of the microbe. So there is persistent of inflammation. Then sometimes these enzymes are released in excess. So when they are released in excess, they are responsible for tissue damage that occur in large area. Examples are acute respiratory distress syndrome, glomerulonephritis. The smaller granules or secondary or specific granules contain lysozymes and phospholipase A2 enzyme. They are responsible for degradation of the bacterial cell wall. Lactoferrin bind iron. They limit bacterial cell growth. The vitamin B12 binding protein is also present in these small secondary granules, and this regulate bacterial cell metabolism. Matrix metalloproteinases such as collagenase or type 4 collagenase specifically, they break down the basement membrane and they are responsible, all these secondary granules or small granule enzymes are responsible for degradation of the microbe and necrotic tissue. They facilitate neutrophil migration by remodeling in remodeling of the extracellular matrix. In chronic wound healing delays, there is excessive matrix breakdown. That means the overactivity of these enzymes that increases the matrix breakdown that delays this wound healing process. If the collagenases are overactive, there is increased breakdown of the collagen, one of the major protein in the structural proteins of the body, and it is implicated in periodontal disease. Then there are tertiary granules in the neutrophil that are also termed as C granules. These C granules contain gelatinases and cathepsins that degrade extracellular matrix for the facilitation of cell migration. There is also presence of urokinase type plasminogen activator in these type C granules of the neutrophil that promote fibrinolysis and tissue remodeling. These enzymes of C granules, they enable neutrophil migration through basement membrane, and it prepares injured tissue for healing. When there is defect in neutrophil migration, uh, it it will reach to the site of injury late, and this is termed as leukocyte adhesion deficiency. Then excessive protease activity can lead to tissue destruction as seen in chronic inflammation of the COPD and rheumatoid arthritis. Macrophages or large eating cells, they are of two varieties. One is pro-inflammatory phenotype and it is termed as M2, while another is anti-inflammatory, that is M2 macrophage or alternate type of the macrophage. The pro-inflammatory type of the macrophage, they have inducible nitric oxide synthase as we have seen earlier, and these produce nitric oxide for the microbial killing. There is presence of pro-inflammatory cytokines such as tumor necrosis factor alpha, interleukin 1 and interleukin 6. These cytokines are responsible for amplification of the immune response. There is also production of reactive oxygen species and lysosomal enzyme that are responsible for killing of microbe and degradation of the tissue. So the function can be summarized as microbial killing, cytokine production, and activation of the immune response. If the pro-inflammatory macrophages are overactivated, they are going to secrete more of these cytokines, and they are responsible for the destruction of the tissue as seen is tuberculosis granuloma. The macrophages are so much changed in tuberculosis that they attain the form of epithelial cell due to their secretory activity, and therefore they can be termed as, or they are known as, epithelioid cells like epithelial cells. Then persistence of tumor necrosis factor alpha production by these macrophages can lead to cachexia that is seen in chronic diseases. The other type of macrophage is M2 macrophage, alternate macrophage, that has got anti-inflammatory function and has got different set of enzymes. There is arginine that converts arginine to ornithine instead of nitric oxide. There is transforming growth factor beta that promotes tissue repair and fibrosis. Interleukin 10 is also in this battery of enzyme that suppresses inflammation. So overall, all these lead to tissue repair and remodeling, and there is resolution of the inflammation. Clinical correlation of this anti-inflammatory or M2 type of the macrophages, there is fibrosis and excessive tissue repair when they are activated more, and as seen is chronic liver disease and cirrhosis. And when there is deficient M2 activation, there is delayed wound healing as seen is diabetes mellitus. So this inflammatory environment is a proteolytic environment where there is a lot of degradation is going on. To control and limit and confine this activity, body has got some proteinases inhibitor. These proteinases inhibitors are there to control or to regulate the proteolysis that is going on over there. There are many of these. One such inhibitor is alpha 2 macroglobulin, that is a broad spectrum inhibitor of all classes of proteinases. It traps and inhibits proteases in the plasma, and hence it prevents excessive tissue degradation. Deficiency of this proteases will contribute to overwhelmed inflammatory disease and excessive proteolysis. The serpins or serine protease inhibitor is another class of the inhibitors. These act on serine proteases such as thrombin, trypsin, and elastin, and regulate coagulation, inflammation, and fibrinolysis. They prevent excessive breakdown by the proteases, and this mutation in serpin is responsible for the effect uh, that are seen in emphysema and the liver diseases due to deficiency of the enzyme alpha-1 antitrypsin that regulate the activity of proteases and antiproteases. Alpha-1 antiproteases or alpha-1 antitrypsin or alpha-1 anti-chymotrypsin is it targets human leukocyte elastase or cathepsin G and protects lung and connective tissue from neutrophil elastases, and it limits by working by confining the elastase activity. It limits the damage that is done by these inflammatory mediators. So the examples are alpha-1 antitrypsin deficiency and COPD. LFN or secretory leukocyte proteinase inhibitor, they target proteinase 3 and they target proteinase 3 and they regulate neutrophil elastase in mucosal tissue and protect against excessive inflammation, and their deficiency is linked to lung disease, chronic lung disease such as cystic fibrosis and bronchiectasis. The plasminogen activator inhibitors, uh, the target enzyme is urokinase type plasminogen activator, and it regulates fibrinolysin, prevent excessive plasmin activation and bleeding. When this PAI and plasminogen activator inhibitor are increased, there is thrombosis risk. There will be formation of deep venous thrombosis and chances of stroke. When these plasminogen activator inhibitors are decreased, there is tendency for bleeding disorders. Another inhibitor of proteolysis are tissue inhibitors of metalloproteinases that are termed as TEMPs, tissue inhibitor of metalloproteinases. The target enzyme is matrix metalloproteases that are MMPs, and it balances extracellular matrix remodeling and healing of the wound, and they prevent excessive matrix degradation. When this activity by tissue inhibitor of matrix metalloproteases is increased, there is fibrosis and severe organ scarring such as seen in pulmonary fibrosis and liver cirrhosis. When this TIMP activity is low, it will lead to invasion of the cancer or metastasis. So to summarize the intracellular killing and degradation, this step is the process of breakdown of engulfed microbe inside the phagolysosome. There are various mechanisms by the production of the reactive oxygen species. There is NADPH oxidase. This oxidase enzyme is disassembled present within the cell in the disassembled or unorganized state. Whenever there is recognition of the foreign particle or ingestion or engulfment of a microbe, it is formed on the surface of the phagolysosome, and it forms superoxide anion. This anion is converted to hydrogen peroxide by the action of superoxide dismutase. When myeloperoxidase enzyme that comes from the alpha granule or large granule of the neutrophils, it converts this hydrogen peroxide by the action of halide or chlorine into hypochlorous acid that has got potent microbial activity, and hydroxyline that is also microbicidal. And hydroxyline is formed through the Fenton reaction. Then reactive nitrogen species are also formed. Inducible form of nitric oxide synthase form peroxynitrite, and this is toxic to microbes. Then there are many lysosomal enzymes that are lysozyme, defensins, lactoferrin or protease, and all of these lysosomal enzymes, they are responsible for the damage. If there is deficiency of NADPH oxidase system, this will lead to chronic granulomatous disease. There is impaired production of the reactive oxygen species and susceptibility to catalase positive bacteria such as Staph aureus and Aspergillus. So neutrophilic extracellular traps are extracellular fibrillar network that are released by neutrophil, and they trap microbes and concentrate antimicrobial substances. The composition of these neutrophilic extracellular trap is nuclear material, that is DNA and histone, and there are many granular proteins. They are antimicrobial peptides and enzymes. The triggers for the formation of these neutrophil extracellular traps are inflammatory mediators such as cytokines, interferons, chemokines, complement proteins, and reactive oxygen species. These neutrophils extrude chromatin and they form a viscous meshwork, and in this meshwork the bacteria are trapped, such fungi are trapped, viruses are trapped, and they prevent spread of microbes, and they concentrate antimicrobial enzyme and peptides at the site of infection. The formation of net leads to loss of neutrophil nucleus, resulting in cell death, and this specific type of the death is termed as netosis. It is one of the form of apoptosis. This is termed as netosis, neutrophil extracellular trap osis. These neutrophilic extracellular traps have got some role in sepsis. The nets circulate in the bloodstream during sepsis, and their formation in the blood is linked to the activation of the platelets. So they have got some protective role as well as some pathologic role. Uh, they enhance the microbial killing and prevent dissemination of the infection, and they also contribute to autoimmune diseases such as lupus by exposing nuclear antigens to the immune system. So these nets represent a double-edged sword. They enhance the microbial defense but may also derive autoimmune pathology. So the neutrophil extracellular traps are seen in blood during sepsis, and they are also source of nuclear antigen in the systemic autoimmune diseases. So here in this picture, you can see that the neutrophils are present and these are healthy. Their nuclei are stained red and cytoplasm is stained as green. And in this picture, there is a release of nuclear material outside, and fibrillar meshwork is also gone outside. Two of the cells have lost their nuclei and they are forming these extracellular traps within this area. This one is the formation of the nuclear extracellular traps. The picture over here, it is showing that there are numerous microbes and these microbes are trapped inside. These microbes are staphylococci and these are trapped in this extracellular meshwork of the neutrophil, and this is seen by electron microscopy. The fourth and final step of the phagocytosis is extracellular release of granules that result in tissue damage and inflammation. And this tissue damage and inflammation can harm host tissue. This is done by reactive oxygen species and proteases, cytokine release that amplifies this reaction, and neutrophilic extracellular traps that trap and kill the microbes but can also bring the collateral damage. So in septic shock, there is excessive neutrophilic activation and there is widespread tissue damage that leads to organ failure. In autoimmune diseases such as rheumatoid arthritis, there is chronic neutrophilic activation that leads to the effects. Thank you for watching this video. If you have any question, please write in the comment box. Thanks.